All right, hey everybody, we're back. I just got done with an awesome clubhouse on Twitter. So for those of you watching the video, look at my tweets on Twitter and try to, if you want, we have this awesome clubhouse, which is kind of this Android app. We have a proteomics chat every 2 weeks with Ben Neely and I and 10 to 15, 20 other people that show up and we ramble for an hour. So anyway, I'm done doing that. It was a great one. So let's get back to what we're doing. We're looking at the post-analysis of this 50 nanogram DIA QC runs. So instead of the analysis tab, we go and look at the post-analysis. And the first thing we see is number of identifications. It tells you the precursors. We talked about that earlier, the modified peptides, the protein groups, proteins. You can see there's two numbers here. And I think this is the number of, it splits these up into average profiles, sparse profiles, and full profiles. And so that's an important point that's not well documented in the manual, or maybe they changed it, but I didn't understand what it was the first time. So average is the number of, I think this is found in greater than half your samples, I think, or half or greater. So for example, there were 5,000 proteins that were found in these experiments with over half the samples. Sparse profiles, I think, means less than half. So those were actually more, which I guess you would expect these numbers to increase as they are found in less samples. So, for example, there's 5,160 proteins found in less than half the samples, and on average there were 5,000. Full profiles mean that they were found in every sample, so that number goes down, of course. So we saw 4,100 approximately 4,100 proteins found in all of these samples, and the same can be said for peptides and modified peptides and precursors. So data completeness is, this is an interesting proteotypic peptide. So a lot of people use proteotypic, the word proteotypic differently. Unfortunately, it's defined differently by different groups. I think this is the standard way to define it, I think, or maybe it's the most current way to define it. But the way it's defined here is that proteotypic peptides are peptides that are specific to a protein. So this peptide was only found in one protein. I believe that's what the definition Spectronaut is using. So for example, we talked about one hit and two hit wonders, or we filtered away one peptide and we just kept two. But there are certain peptides that are only specific to one protein group and some that are specific to more than one. We talked about this with protein inference too. But I believe that's the way the definition's used here. So this is a peptide that uniquely identifies a protein, I believe. Okay, average number of miscleavages, 10%. That's pretty good. Data completeness. This is that's a good question. Let's see what this says. The percentage of precursors individually identified in a sparse profile matrix. Oh, that's interesting. Okay. So you can see how this goes up from 25 to 90 minutes. So might be a good example of why to use a longer runtime. Recovery. So the percentage of precursors, so this seems to be all in the precursor level, from one experiment versus the library. And so basically what Spectronaut's doing is it's making a library out of all of the runs and telling you how much of it's recovered in each individual condition. So for example, we're recovering 82% of the precursors in 25-minute runs, but we're recovering 85% in 90-minute runs. That doesn't seem much difference to me. This is the CVs across our different run lengths. You could see they're a little lower in the 60-minute runs, higher in the 90. That's interesting. So you would expect the more points across the peak you have, the lower your CVs are going to be. But there's other things I take into account CVs. Peak capacity, I guess you can put your little mouse over that. Does that tell you how peak capacity is measured? Doesn't look like it. That is defined in the manual somewhere. XIC width, this is the amount of the time domain we're using to extract the signals. And you could see this going up under a 90-minute run, and you would expect this because your peaks are going to be wider in a 90-minute run. So that makes sense. I don't know why these say not a number, but we'll go into average data points per peak later. Run identifications. This is kind of a useful thing. You could see there's something wrong with this 45-minute run. There must have been something wrong with the autosampler or the column. It's strange because we'll have to look at the run order when this was done. I don't think it was the last run. It wasn't, because I did 25, 45, 60, and 90. So whatever happened to this one might have been like an autosampler issue or something. And then you can look at the number of precursors, modified sequences, peptides, protein groups, and proteins, and how they differ across the different runs. You could see the 25-minute runs are not that much different than the 60 and the 90. So keep that in mind. So let's see. Data completeness. Oh, these unique identifications are useful. So if you see a big red bar on your run, that means these identifications were only found in this sample. So we don't see a lot of that here, which you would expect. But if you're doing like an IP, for example, and you have a control versus experimental, you might find a lot of this bar being red because they're only found in one condition. Okay, data completeness. This kind of shows you what we looked at under the analysis overview text, but it kind of shows you a bar graph of it. So out of all of the runs together, we're recovering about 95%. So we're able to quantify, I think, about 95% of the data. And I think you can group this by modified sequence, protein group. So I would expect this to be more uniform on protein group. Oh, let's see, that's interesting. So you can kind of see the differences in the proteins. So like full profiles, that's interesting. Anyway, I have some theories about why this is, but you can change the group to strip sequence, precursors, kind of give you an idea of how consistent your data is. It's kind of interesting. This is your missing values. So this is the number of times things are missing. I believe this takes into account imputation. So like even if you impute it, I think this is before imputation, I think, but I'm not entirely sure. So we have 7 runs here. Is that 1, 2, 3, 4, 5, 6, 7, 8? We have 8 runs. And you can see there are 25,000 precursors that are not missing any values. And this just means it wasn't found in 1 run, wasn't found in 2 runs, wasn't found in 3, 4, 5, 6, and 7. So this was the difference between, if you subtract these numbers, these are the ones that were only found in 1 run. And you can change the group by again by using our secret right click. And we can change this to like protein group, for example. And you can see most of the proteins, as you expect, would be found in all of the samples because this is just a QC. Let's see, you can do by like strip sequence. That might be interesting to show. That's kind of cool. But we're going to go back to precursor again. And remember the difference between a precursor and a peptide sequence is peptide sequences can have more than one precursor based on like chart state. Okay, rank protein groups. This is really interesting. We did differential expression, so you can see it's kind of a blurry mess. You can zoom in if you want, and it makes things more easy to read. Right-click, set scale to default. I am going to unclick highlight candidates. So because we did differential expression, so basically let's turn this off. So we'll explain this. We'll explain this graph. So this is roughly the dynamic range between your most abundant protein, which would be here, and your least abundant protein, which would be there. And we can show point info. and then it'll show you what this is. So this is roughly the most abundant protein in our sample, and this is roughly the least abundant protein in our sample. If you really wanted to make it more accurate, you could like turn on IBAC, which is essentially this number normalized by the molecular weight of the protein. So for example, you would expect on average larger proteins to give you more peptides and more signal than a smaller protein. So for example, histones are really small. So they might, even if you had a ton of histones, they might show up here. Whereas if you change this, what would be nice is Spectronaut had a little check mark to say IBAC, to turn on IBAC on or off, to normalize this by molecular weight. And then usually when you do that, you see your histones jump way up here because they're usually abundant, but they're small. So when you just measure the total signal without normalizing them for molecular weight, they kind of end up here. But the proteins that are differentially expressed will be in red if you've done that already, and we have. So like, for example, this one's shown to be changing between our runs with their different gradient lengths. And so you can look at those if you want. But essentially, this is a measure of your dynamic range of your samples. So remember, this is a log 10. So we have like 4.8 to go down to 0.8. So we have 4 orders of magnitude, so 10,000 times. So basically, we're able to tell the difference between the most abundant protein, the least abundant protein, and those can differ by 10,000. So that's pretty good. But I love this graph. I think it's really interesting to look at where your proteins lie on this graph, especially if you have like a viral protein. Like we did a COVID project, and so you can kind of tell where the spike in membrane and nucleocapsid proteins are. But you can also do custom selection. So if you go custom selection, you could search for a protein. Like, let's say we wanted to see where EF2 was. You could just search for your protein here. Here's EF2. You can say user selection and then hit OK. And then you could see it turn up in blue right here. So that's really cool. So custom selection. Like let's say you have bait proteins, right? And you're doing an IP or something, or you have your proteins that you think are super important. You can select those, like search for them, hit custom user selection, hit OK, and they will show up as blue dots on this graph, which is a great way to tell, is your protein one of the most abundant proteins or is it one of the least abundant proteins in the sample? And so usually your quantitative accuracy is going to scale, is going to be influenced by the intensity of the signal. So you may be able to, you're probably not going to be able to quantify these as accurately as you can on the top. So keep that in mind. But I love this graph. This is one you should always look at. All right, digestion efficiency. This is how well our enzyme worked. Promega did a good job, mostly highly specific. So that's good. CVs are good. Let's see, where is CVs below X? Oh, that's interesting. Why is it only showing me the 25-minute runs? I don't know. But let's ignore that for now. Normalization. So this is the original signal of your samples, and this is how they are normalized. From this, you wouldn't entirely see there was something wrong with that 45-minute run, but there definitely was, because it identified one run identified way less than the other, even though the signals look similar. So for example, you could see how the 25-minute runs have an overall less response than the 45s, and the 60s kind of level out with the 90. And you kind of expect this because we're kind of pushing things a lot quicker. You're not going to have as much time to measure the signal, maybe. That might be one reason. I'll have to think about this more. This is complicated. Anyway, you could kind of see the difference between the original and the normalized. And what you want to watch out for here are outliers. So, like, for example, if these are all on one line and you see one that's really lower, and we see this a lot, not a lot, but we see it more frequently than we'd want, that means there's something wrong with this sample. Either it was digested differently than the others. It could be sample prep before we get it. It could be a mass spectrometry problem. But definitely look out for outliers. Spectronaut is only able to correct it to a certain amount, so just be careful. Okay, the tick overlay plot. Oh, this is pretty, huh? So this is how your tick looks, your total ion chromatogram over time. And you can see the 25-minute runs, 45-minute runs, which are these two, our 60-minute runs, and our 90-minute runs. So these are all the peptides, how they get spread out over time and retention time. And you can see the 25-minute runs did pretty good at the beginning. And so remember, you're going to pay by time, unfortunately, so I have to charge you. I know, they're mean like that. So anyway, we've seen the 25-minute runs did a pretty good job. Sample correlation, that's cool. So you can see how the samples correlate with the other samples. So you would expect the ones in the same gradients to correlate with each other more, and they do. So for example, this 60-minute run correlates well with this one, right? So that would be this one and that one. So correlation of 1 if they're the same sample. And this one has a correlation of 0.996 between the 60-minute run 1 and 2, but only has a correlation of 0.89, which is still pretty good if you can compare a 60-minute to a 25-minute. And these look really bad, which you would expect, right? So the 25-minute runs only have a correlation of 0.83, which is actually really good between the 90-minute runs. So that's useful. I could probably write a paper out of this. I actually don't know what explain tick plot does, but it is blank. Apparently the expensive tick plot function didn't do anything. Maybe that's a bug. Maybe it's another reason why. It's the first time I did this. I don't know why it's blank, so maybe I'll figure that out. Coefficients of variation. This is a very interesting plot. It defaults to precursor, I think. You can hit your secret right click and go precursor, select peptide. Let's just do peptides. Did that change? So you can kind of see how consistent we are across runs. So the lower the CV, the better. And you can see it's highest with 25-minute runs. So you sacrifice quantitative reproducibility for time. So for example, you can see how if you zoom in on the 90-minute runs, 60 is actually a little bit better than 90. But you can see how it's worse with 25-minute runs. And what's interesting is if you look at this data, reset the default, you can see how the curves aren't exactly what you would expect, right? So like, for example, it's kind of hard to get a median out of this. I mean, when you say these are not exactly Gaussian shaped curves, I don't know if the median actually even makes any sense when curves look like this, but you can see how the CVs can vary greatly by some precursors versus others. So some have a very high coefficient of variation between the quantitative measurements and some do not. So this is actually one of these graphs that's important to look at the shape of the curve because the shape can change. Here's the box plot of the data. So you can see how it's higher with the 25-minute runs and lowest with the 60-minute runs. Data point per peak is what I like. And you can see how we found an issue with these 25-minute runs, which we subsequently fixed. So this is the number of times we can sample this, the average peak as it comes off and on the column. So it has to do with the speed of the duty cycle of the mass spectrometer, the windows we use, the peak shape of your peak. For example, if you have really wide peaks, you're going to be able to sample it a lot of times. You have really narrow peaks, you're going to be able to sample it not so many times. And you can see for the 25-minute runs, we're only sampling at median of 3.2 times with, it seems like most of them are, well, that's the median. But you can see the shape of the curve. So that's why a lot of these data points look like triangles because the average is only 3. And look, some are less than 3. So I think, so anyway, that's why we were not able to get as good quantitative accuracy. But surprisingly, the CVs weren't that different, right? There was a 13% CV of the 25-minute runs were only a 10% CV or a 9% CV for the 60s. So based on this one, you would expect them to be, the CVs to be a lot different, but they're only like 4% different. So I don't quite understand that. So like what the take home for this is, and this is kind of geeky mass spec info, is that a lot of times in these mass spec method papers, you will see like people quoting the median data points per peak, right? But if you actually go back and look at these curves, you realize they look really funny. Like they're not Gaussian a lot of the time. So I mean, this might be right, but I don't know, man. Does that make any sense to look at the median when a curve looks like this? I don't know. But anyway, you can definitely see the difference between a 25-minute run and a 90-minute run. You can see we're doubling the amount of peaks, data points per peak on the 90-minute run. So you'll pay for that quantitative accuracy. And we've subsequently fixed this. On my Spectronaut video, you could see it. And I'll probably show it later. But we've upped this for the 25-minute run to be around 5 or 6 points per peak now by increasing the window size. Okay, bend identification. This is not... these are actually interesting. So this is the RT. They call these RT buckets, or retention time buckets. So that's if you split your gradient length into time, you could kind of see where most of your IDs are, right? So what you don't want to do is have, this will show you, for example, like on this yellow one, which is the 45 minutes, how we're identifying less at the end of the gradient than we are with the 60 and the 90 and the 25. So at the end of the gradient, the 45 minute is not doing so good, which means we might have to just adjust the 45 minute. And I think this is the log 10 quantity based IDs grouped by condition, log 10 quantity buckets. So for example, this is the signal. So like number of IDs with a high signal versus low signal, I guess. And this is just your mass overcharge grouped by condition. I don't know why most biologists wouldn't need this, but maybe for an analytical chemist improving their methods, might be important. Or if you're doing cross-linking or something. Bend coefficient of variation. Let's see what we got here. Oh, that's interesting. Remember how we saw one of those 45-minute runs is an outlier, even though the normalization looked pretty consistent, right? Although this is a log 10 response, so maybe that is a significant change. But you could kind of see how your coefficient changes. CVs bucketed by IRT, which is the same as, which is kind of the normalized, or the IRT space for retention time. This is interesting. So there's something going wrong in this hydrophobic section of this 45-minute run. So the first part of the 45-minute run was fine, it's telling me. And then once it got halfway through the run, something terrible happened with the 45-minute run. So that this is telling me half the 45-minute run worked, half of it didn't. And so you kind of want to look for outliers like this. It should be fine. It should be the same. When you see stuff like that, that's a red flag. That's telling you something's wrong with this run. Let's see, label-free scoring histograms. This is if you want to get into something geeky about how the scores are done, the decoys versus the experimental conditions. These are how the posterior error probabilities and the Q-values are calculated. So for those of you that are into that thing, it's very useful. I've never really been a biologist where this is interesting. But if you want to know how the tests work and how it's segregating decoys, so a lot of things in proteomics feed it false data. So like the decoys are fake signals or fake peptides, and they try to extract the signals from it. And this is try to separate the good data or things that are known bad from things that we think are good. So it's an estimate of the quality of the error in our data, and so it's filtered here. So like, for example, what this is telling you is, these are at 1%, you're still going to have some data that gets through, but at 1% of the data. Protein FGR, this is kind of similar, and you can see that for the same for each run. So these are the decoys, this is the non-decoys, and we try to calculate where the example is. And you can see if this might be an example of, you can filter by score. It's an interesting graph. I don't really know what this means. This seems a little into the red for me. I would think it should be out here, wouldn't you? I don't know. I'll still have to figure out what that means. This is always good. I always like to look at the p-values. So I believe this is the p-values for the scoring, not the differential expression. So we see a nice p-value, nice bar here, which is what we should expect. You could ignore LFQ bench, so this is kind of like a geeky thing. So if you have more than one species in your experiment, you can separate it based and determine the differential expression based on different species. So usually there's a paper called LFQ bench. This is what they use to validate this kind of approach. So like if you take yeast and bacteria and you mix them at a 1:4 concentration, you can see kind of what the graph should look like. And you can look at these LFQ bench papers. So all right, differential expression candidates. So this is kind of a cool graph. So we set 25 as the reference, remember in the other video. So it's going to compare all of them against 25. So for example, and if you click on these will change. So you can see, for example, this protein was differentially expressed between the 25 and 45 and 60 and 90, even though it's the exact same sample and the exact same amount, but because we're doing something silly and comparing it against different gradient lengths, it thinks it's differentially expressed. So, if the normalization and the IRT and everything was perfect, it shouldn't be, but nothing's ever perfect. It's actually way better than I thought it would be, surprisingly. So anyway, for whatever reason, this protein has more of a response at 25 minutes than it does at 90. Why is that? I don't exactly know. I've been trying to figure this out. If anybody has any ideas, let me know. I think it has to do with the number of peak shape, maybe number of peptides. I don't exactly know. And we can find opposites too. So let me explain this. There's two graphs we see here. So the red one at the bottom is the signal, the MS signal. That's the signal on the precursor. The green bar is the signal we get from the fragments from the precursor. And you can see how the green bar is always below the red. And that's just a mass spectrometry thing. So like the precursors are always going to have a higher intensity than the fragments. This is especially the case on timsTOFs, where the signal, MS signal, can be quite high, and the MS fragments can be quite low. And then what Spectronaut does, and there's a paper on this, I think it's in the manual, is it combines these, but it doesn't in a way where it scales it. So if you just combine them, you would just get the signal of the MS, and you don't want that. So you could, to make use of both the MS signal and the MS signal, you scale them in a way to give you the quantitative profile on top. So, and this is a log 10 response, and I think you can say do not use log 10, do not use the secret right click. And as a somewhat of a biologist, I like this one better because you can really see the difference between the if you're using normal scales. So like for example, this one's way higher in the 25s than it is in the 45s and 60s. This one's actually the opposite. So it's actually, this protein has a higher response in the 90 minutes than it does in the 25. And this kind of tells you what way it's going. So for example, if it's higher in the 25s than the 90s, for example, it'll be red. And if it's the opposite, it will be green. So you can kind of see that here. which is interesting. So this is telling you it's comparing the 45 and the 25, and it's more in the 25. So 45 is less. So down-regulated in the 45, which is what it's showing you. This one is upregulated in the 90 over the 25. Very interesting. So you can tell there's 20, we found 2,974 proteins significantly changing, I think, different between conditions. You can see there's 15,000. That's because we're comparing 3 different conditions. So that doesn't actually mean 2,900. We could figure this out in the report. So you can also filter up here. So they do default. So you can see there's a default here of the average log-2 ratio to 0.58, which is 1.5-fold change, right? Approximately 1.5-fold. So you can change this. So let's say you want to look at only things that are 2, like really different. And that changes here. So now we can find things that are really changing between the 2. And you can make these whatever you want. Like 4. Let's see. No, wait, look at that. Whoa. So remember, that's like a fold change of like 16, right? So let's see, we do like 3. You kind of change. You're looking for the things that are really different. Like, look at that. That's interesting. And the size of this, what does the size mean? Size must mean something. So it defaults, just remember there are default filters here. Let's see what other Q value. So we filter on the Q value and not the P value. Q value of 0.5 for the differential expression, so 5%. If you want to do this to 1%, you can filter away some of the things, but it defaults to 5%, which I think is reasonable for a p-value for a t-test. So that's kind of the default. I still have to understand what ratios mean. Maybe Spectronaut will tell me, but I think this is ratios between the peptides that it's using. I think this has to do with the LFQ ratio, so LFQ uses ratios. I believe that's what this is. It's not really defined anywhere I can find. So uniprot IDs, genes, protein description, protein names, unique total peptides. You shouldn't find any ones because we filtered them away. You can sort here, and you can see the lowest number is 2, highest number is 63. It tells you the ratio. I believe that's the log-2 ratio. Condition name, numerator, denominator, gene ontology, molecular function, that kind of thing. You can look at the p-value histogram. Remember, this is a different p-value histogram than the one here, because this is just the one above here is trying to figure out what signals are real peptides. This is actually a p-value histogram of the differential expression. You can see it's okay, right? Not as sharp as it used to be. But it'll work fine for a multiply tested corrected, like a BH correction, right? So you want to watch out for your p-value shapes when you're doing multiple testing correction. All comparison histograms, that's interesting. So this is the average log-2 ratio across all comparisons. You can see it's kind of, the median looks like it's not exactly 0. You could change the bins. Principal component analysis, which is interesting. So this is how well you've done on your experiment. So if you have different conditions, obviously you want your PCAs to be well separated. Sometimes they are, sometimes they're not. And you could see the 25-minute runs are a lot different than the 60s and the 90s, which are similar. And you could see there's one 45-minute run that something happened, and you could tell there's something wrong with this run. And to figure out what this is, you can do our magic right click and click on show point labels. And then you could tell that this one is the 45-minute run replicate 1. And so you could go back to your analysis to figure out what run that is. So like if this were me, I would throw this out and do it again. But this is also telling me that the 60-minute and the 90-minute runs are more or less the same. So there's maybe no use to doing a 90-minute run on the timsTOF And I believe that's kind of my conclusion here. Okay. All right, geo-enrichment. This is actually hard for me to understand sometimes. But these are the different conditions. So we're looking at 45 minutes versus 25 minutes. The dot indicates the size of the dot corresponds inversely to the p-value. So the larger the dot, the better the p-value, I think. The lower the p-value. And the color indicates the direction of enrichment. Red, underrepresented. Green, overrepresented. So what this is telling me is that this gene ontology membrane is underrepresented if you're comparing the 25 to 45. So I don't know, membrane proteins not doing as well. This all doesn't make any sense because this is like the same sample run just on different gradients. So honestly, this makes no biological sense whatsoever. But if you're doing a real biological experiment, it should, I would think. So you can tell, for example, if your membrane proteins are different across your different experiments, or you can look at something like RNA binding. So this is actually really useful, is if you have different profiling conditions, and this tells you that your RNA binding proteins are differentially regulated between your different conditions. But for this experiment, it makes absolutely no sense whatsoever, and I have to think about why it's showing me this. Like, obviously, it makes no biological sense because it's the exact same sample, just run on different gradients. But if you're actually doing something reasonable, like a different biological experiment, this would be useful. Okay, so I don't understand gene ontology clustering yet. Maybe if someone figures it out, you can hit start clustering, and it will do something. I think it clusters the data from this together, but I'm not exactly sure. Volcano plots are useful. You can see, you can kind of see between the 25, we'll look at the 25 and 45. What this is telling me is that, let's see, what is this telling me? That a lot of these proteins have less abundance in the 25 than the 90, which you would expect. So remember, when we run your samples for real, we run the same gradient over all your samples. Anyway, that tells you what on the bottom, that's the full change, log 2 full change. And on the y-axis is the p-value. So you look for things with high full change with a high p-value, low p-value. So this is the inverse, the log 10 of the p-value. So this would have a good p-value that's differentially regulated. I wish you can click on these. So Spectronaut, maybe in the next version you could click on this and go back to that tree view under the analysis. That would be useful. Let's see. And you can save these, right-click, save image as. You could do a custom selection. So again, like if you wanted to see where this protein was in the volcano plot, you could, and it's right there because it's not changing. Heat map. So this is the heat map. What you want to see is your different conditions clustering together on the bottom. It's kind of like a PCA that way because they're clustering. So your run should cluster based on conditions at the bottom, and your proteins will cluster based on how they're changing. So for example, you might see these here. You could see this 45-minute run here is white, which means they're missing from this one. So there's something wrong again with our 45-minute runs. But even in our 25-minute runs, you can see how I have more variation here. There's some missing between one run and the other, where this is less likely the case in the 60. And you can see, you can zoom in and kind of see how these are different. Like you can see this one's missing in this 90-minute run, where it's not missing on this one. And remember, this is because we've turned imputation off, but if you go back and turn imputation on, you might not see these. I don't know, we can go and test it. Restore to default. Okay, so that's all I'm going to talk about now, and then the next one, we're going to go over the report, and I think we'll be done. All right, so thanks for sticking with me for one more time. I think we'll have one more to go, and then maybe a follow-up after that. All right, well, thanks, everybody. I'll see you when you click on the next link.